Okay, so good evening, guys. So I'm Rishabh, and today we'll be starting with the week 5 of the course Host Pathogen Interaction by Professor Himanshu Kumar from ISER Bhopal. And in this week, we'll be majorly discussing about the innate immunity. Okay, so and the major components that takes place inside the body include are involved in the innate immunity. Okay, so if you guys have any doubt, you can ask me in between. So let's start. Okay. So starting with the innate immunity. So innate immunity basically started nearly around 700 million years ago when the sponges, okay, the sponges, the basic animal kingdom organism arose. So as you guys would know that sponges were the first phylum of the animal kingdom of the invertebrates. Okay, so the innate immunity started with these sponges, and then it came the antimicrobial peptides, the phagocytosis, complement system, the toll-like receptor, and then after that, nearly 450 million years ago, there was the development of the adaptive immunity. The basic adaptive immunity that started with jawless fishes. Okay, so from adaptive immunity, the trend started for MHC, immunoglobulins, the T cell receptor, RAG, and the lymphocytes. Okay, so these all are associated with the lymphocytes. And now, as humans, the most developed kind of the animals, so now we have the most advanced uh, immune system. Okay, so this is the major evolutionary pathway of the immune system. So now we'll start with the innate immunity, that is the first line of defense. Okay, so it is basically the first line of defense that takes place inside the body and that helps for all the pathogens or any of the parasites that try to enter the body. So the innate immunity actually helps to barricade all these things and try to protect the body from all the particular pathogens, allergens, and many other things. Okay, so there are certain components of the innate immunity. First is the physical, mechanical, or anatomic barriers. So this correspond to basically the membranes or the surfaces. Okay, so membranes in terms of mucous membranes as present in the gut or the respiratory tract, or surfaces like the major one, skin. Okay, then we have the biochemical or chemical barriers. These include antimicrobial peptide or proteins like mannose binding protein, CRP, APP, serum amyloid A, alpha 1 antitrypsin, ceruloplasmin, human beta defensins, lysozymes. All of these are the antimicrobial peptides or the proteins. Then we have complement system, the microRNAs against various viruses. So, talking briefly, microRNAs are basically components or small RNAs, okay, that are helpful in regulating the expression of various virus related proteins. Okay, so the basic funda is if the replication or the translation expression proteins are not produced by the human body for the viruses, then the viral replication will not take place, and hence it will not be able to divide itself in the body. Okay, then we have the microbial or microbiological barriers. So in this case, what happens, as you guys would know, that in our human body, there are more number of bacterial cells that are present as compared to the human cell. Okay, so because of this bacteria present inside the body, there are various functions that takes place. For example, digestion of various foods, because all of the things are not digested by the human body. So the bacteria help in digesting the food also, as well as it helps in protecting the body from certain organisms. Okay, then we have the cellular component, that is the immune cells and the non-immune cells. So immune cells including NK, natural killer cells, the macrophages, the dendritic cells, and the cells that express the pathogen receptors. Okay, so these receptors basically help to sense any of the products of the metabolic pathway or any of the biomolecules, so that they can actually detect the pathogens. Okay, so here is the distinction. So we have the skin, the first line of defense, then we have gut, the lungs, the respiratory tract, then we have eyes, nose, and the oral cavity. Okay, so all these things are the most exposed part of the body. Okay, so this is your skin that is present. Then you have your gut, because the outside food that goes directly into the gut, the outside air goes directly to the lungs, and eyes are the direct contact, nose through which you breathe, and oral cavity through which you eat. So all these things are the first line where you are actually directing directly are in contact with the environment. Okay, and these all the layers are made up of epithelial cells. So this is the most common thing that is present. So now we'll see the mechanical barrier that is present. So all the epithelial cells are joined by the tight junctions. There are no loose junctions in between the membrane, so that anything cannot enter freely inside the body. Then for the skin, we have the longitudinal flow of air or fluid. Same for the gut. Then for the lungs, we have movement of the mucus by the cilia. Okay, and for the eyes, nose, and oral cavity, we have tears. Tears also help in the mechanical protection and the nasal cilia. Okay, for the chemicals, we have the fatty acids, the defensin, laminar body, and cathelicidin. For gut, we have low pH enzymes like pepsin. Then we have certain peptides like alpha defensin, regil, and then cathelicidin. Same for the lungs, we have pulmonary surfactants, basically mucus, that helps in free flow and the trapping of the particular pathogens and the dust particles. And the peptides includes alpha defensin, cathelicidin. Then in the cavities, we have enzymes in tears. Okay, so which enzyme is present in the tears? It's the basically hmm, it is the lysozyme. Okay, and in saliva also it's the lysozyme. And then we have histatins and beta defensins as the microbial peptides. Then we have the microbiological barrier, that is the normal microbiota that is present inside the human body. Okay, so these are the different barriers that are present in the different membranes of the human body. Okay, so let's move on and start with the skin. So, skin is basically skin consists of two major layers. First is the epidermis, and the second is the dermis. So, epidermis is the outer layer, and it is tightly packed with the cells. Okay, there are if few pathogens can penetrate these layers, and then the dead skins that we shed. by any of the changing of the seasons. So these actually remove dead skin cells that remove the attached microorganisms. Okay, so here it is the densely packed layer, epidermis. Then epidermal dendritic cells are phagocytic pathogens. Okay, so those are called as Langerhans cells that are present in the skin that helps in the degrading and phagocytosis of the organisms. Okay, these cells extend out among other cells of the epidermis, forming a network to intercept invaders. So the major role is they can actually recognize the pathogens and they can remove them from the skin. Then comes the second layer, that is the dermis. So here we have collagen that makes it stiff. So it gives strength and pliability to resist abrasions that could introduce microorganisms. So it resists for the abrasions that could actually introduce the microorganisms. The perception secrete perspiration secreted by the sweat glands. So the sweat glands are present in dermis that secrete perspiration. So they have salt that inhibits the growth of the pathogen. And then we have antimicrobial peptides like defensins and cathelicidin. And also we have lysozyme that destroy the cell wall of the bacteria. So these are the components of the perspiration that is secreted by the sweat glands. Okay, now we have sebum that secrete sebaceous, or that is secreted by the sebaceous or the oil glands. So this helps the skin in keeping it pliable and less likely to break or tear. Okay, and it has a lower pH, so that helps in maintaining that helps in inhibiting the growth of the bacteria. Okay, because at low pH, that is acidic pH, so the bacteria or any other pathogenic organism cannot grow. Okay, so these are the different layers of the skin that includes dermis and epidermis. Out of those, the this is the close-up structure of the particular skin layer, and this is a watertight lipid layer where we have the laminar bodies, and in these laminar bodies we have the antimicrobial peptide, that is the beta defensin and cathelicidin. Okay, so this was the basic introduction about the skin and different layers and the different biochemical barriers that are present there. So, any doubts about this? Okay, so let's move on to the next part, that is the mucosal surfaces. Okay, so mucosal surfaces are present on any of the lining that governs or that protects from the outside source of the air or food. Okay, so mucous membranes are present in the gut, mucous membranes are present in the respiratory lining. Okay, so the body cavities that are open to the outside environment, the main thing. Then the surface epidermal cells, uh, unlike the surface epidermal cells, epithelial cells are living and closely associated with the lymphoid tissues where antigens are transported via M cells. Okay, so in case of skin, although it was also an epithelial layer, but in that case, what happens? There is clearing of epithelial cells. Okay, so this clearing doesn't take place in the mucosal cells, mucosal surfaces. So here the epithelial cells are considered to be living and they are closely associated with the lymphoid tissues where it can trap the pathogen and the antigens are trapped by the M cells. So as you guys would have remembered, we have studied MALT. Okay, so MALT stands for mucosal associated lymphoid tissue. In this, we have a specified cells called as M cells that have a cavity. And in this cavity, different immune cells were present Okay, so these immune cells were basically macrophages, B cells, and TH cells. Okay, so these cells help in trapping of the pathogens. Okay, then we have epithelial cells that are packed tightly to prevent entry of pathogens, but often only one cell layer thick, so pathogens sometimes breach the barrier. So what happens is, although this is a tight junction, okay, so those are tightly packed, but because it is a single layer, then what happens? The pathogen actually breach this particular barrier. Okay, because of being the single layer. So it can directly enter into the system, crossing this particular single cell epithelial layer. And what happens is, if the layer actually traps certain microorganisms, then continual shedding of the cells, so that if the cells get renewed regularly, the attached microorganisms can easily be discarded from the body. Okay, so this is the main thing that takes place on the skin as well as on the all the mucosal surfaces that they are continuously shedding. Okay, beside producing mucus, mucous membranes also produce lysozyme and other antimicrobial peptides that actually help in destroying different bacteria as well as different microbes that enter through air or food. Okay, every day, an adult human swallows and digests about 1 liter of the mucus. Okay, so this is produced in this much amount that about every day around 1 liter of the mucus is swallowed and digested. Okay, so this is the bronchial ciliated epithelium. So bronchial means respiratory tract. Okay, so as you can see here, this is a blood vessel. So this is the inner lining of the blood vessel and this is the outer lining. So this is the membrane, mucous membrane. This is a goblet cell that secretes mucus, helps in trapping and ejecting microbes. And then we have cilia. So cilia helps in maintaining the smooth flow of the fluid and the air in one direction. So this is the function of the cilia. Then we have gland duct. These are the mucus gland that secrete the mucus, and there are different ducts or the blood vessels that are present in the particular line. Okay, and this is the gut epithelium. Here also we have goblet cells that secrete mucus. Then we have certain cells called as M cells also here. Okay, so M cells are also present here that helps in actually trapping the microbes. Then we have crypts of Langerhans here. So these crypts are present in the stomach and the intestine that helps in absorption of different nutrients. And we have different Paneth cells that are non-immunogenic. Basically, it performs immune functions related to the tissue itself. Okay, so these Paneth cells helps in release of the alpha defensin and the Reggie. And there are certain immune cells that are also present here like macrophages or the dendritic cells that help in trapping the microorganism phagocytosing it. Okay, so this was the basic idea about the mucosal membranes. So if you guys have any doubt, you can ask. Okay, so let's move on. Then we have certain biochemical barriers also that we call it as micro antimicrobial peptides. Okay, so the most important are defensins. So, defensins are basically divided into three types: alpha, beta, and theta defensins. And they are divided into the amino acid sequence that we can see. So, defensins are charged molecules that are positively charged. Okay, and it can easily transfer the membrane because of this nature, and they can create the peptide, the pores in the membrane. Okay, so they are as they can easily transport through the peptide membrane. That means they are both, they have components of the hydrophilic nature, so that they can easily interact with the negative charge of the phospholipid bilayer, and they can create pores. uh, they can create pores so that the membrane is actually ruptured and all the things components can come out of the particular membrane. So this is the basic function basis how this defensin functions. Then each family has its member with distinct activities. Okay, some being active against gram-positive bacteria and the other ones for gram-negative bacteria, while some for the fungal pathogens. Okay, so as you can see here, this is a alpha defensin, and this is the major region that helps in the function, and this is the pro region. And in the beta defensin, the pro region is very less. Okay, so that makes the difference between the two alpha and the beta defensin. Then we also have two more classes, that is regin, uh, lacticidins, and the cathelicidin. Okay, so in case of cathelicidin, we have another peptide that is attached to the molecule, that is cathelin. Okay, so this cathelin is attached to a pro region, after which the cut is made and the pure peptide is formed that functions as an antimicrobial peptide. And in case of Regil, there is a major domain, that is CTLD and the CRD. So that functions as an antimicrobial peptide. Right. Then moving on to lactoferrin. So lactoferrin is basically sequestering the Fe2 plus ions. Okay. So it can easily trap the Fe2 plus ions. So it is a potent antiviral, antifungal, and anti-parasitic nature. So it is broad in terms of its antimicrobial activity. So, in bacteria, it has bacteriostatic properties. To counter its action, bacteria has siderophores. Okay, so because there is always like anti nature peptides also, so if there is lactoferrin for the bacteria, bacteria also had its antagonist, that is siderophores. So in gram negative bacteria, it has bactericidal activity. It binds to the lipid A part of the bacterial LPS. So that is the lipopolysaccharide. Okay. Then we have azurosidin, that is antimicrobial and chemotactic protein, and inactive serine protease. So it is an inactive serine protease that have both antimicrobial and chemotactic properties. Then we have bacterial permeability inducing proteins. So this protein helps in basically increasing the permeability of the plasma membrane. Okay, so that's why the name suggests the bacterial permeability inducing protein. And we have a vitamin B12 binding protein because some of the microorganisms require vitamin B12 also for their growth. Okay, so if you guys have any doubt, you can ask here. Okay, so let's move forward. Then we'll be moving on to the biochemical and the microbiological barriers. So in terms of the biochemical barriers, we have seen about lysozyme, that you guys know. Okay, about it is a enzyme that breaks down the bacterial cell wall. Then you guys just now seen about the antimicrobial peptides. Now we'll be moving on to the third concept, that is the reactive uh, reactive oxygen and reactive nitrogen species. Okay, that increases the oxidative stress. Okay, so we have the reactive nitrogen species, nitric oxide, nitrogen dioxide, and peroxynitrite. So these are synthesized from arginine by NO synthase. Okay, so we have, sorry, peroxynitrite, and this is nitrogen dioxide. And then we have the reactive oxygen species, superoxide ion, hydroxyl radical, hydrogen peroxide, and hypochlorite ion. So these are produced by the NADPH oxidase that was seen in the last class. Okay, so when NADPH reacts with the oxygen, it produces superoxide ion and the NADP plus and H plus. Then superoxide dismutase produces H2O2 plus chloride ions. Okay, and then chloride ions when attached to H2O2 can produce HClO minus, that is hypochlorite ion. So these are the main reactive oxygen species that are produced by the different phagocytes to initiate the immune response. The neutrophils and the macrophages both produce this particular mm, ROS and RNS. Okay, then moving on to the microbiological barriers, there are different classes of bacteria that are present in the human system as an commensalism. Okay, so what is basically commensalism? So generally, the bacteria, if present on uh, any of the humans, okay, so it will be actually example of the parasitism. Okay, so what does parasitism mean? Parasitism mean that the host will provide everything to the particular bacteria, but in return, bacteria will not provide anything to the host. So that means all the benefit that the host provide to the bacteria goes into the pathogenicity, or it will extract only the food from the host. So this is the meaning of the parasitism. Then we have another interaction that is called as commensalism. So in case of commensalism, what happens? We have the host, we have the bacteria. Okay, so host actually gives the place to live and the food to eat to the bacteria. In return, the bacteria helps the host for digestion, for helping in the antimicrobial activity, for protecting from different pathogens. Okay, so this is how both the living beings helping each other for in return of anything. Okay, so this is basically commensalism. So there are different species of bacteria that are present in our gut as well as in our respiratory tract. Okay, so these are lactobacillus and clostridium species that are called as firmicutes. Okay, so this is ubiquitous commensal bacteria that is present in our body. Then we have bacteroides fragilis or the bacteroidetes. Then we have bifidobacterium species or the actinobacteria. Then we have E. coli or the proteobacteria. So these are the major bacterial species that are present in our body as a commensal. Okay, and sometimes these bacteria, although they are inactive and they don't cause any disease, but if their original uh, environment is compromised, then these bacteria can also cause the disease. For example, if you guys have noticed that the in like for example the gut and the this mouth and the this tonsil region if the microbe environment is compromised in any case. Okay, for example, if you drink very cold soft drink or anything, then it actually compromises its environment and they can be activated to cause any of the diseases. Okay, so this was a tiny example that you would have seen in your day-to-day life also. Okay, so these are different pie charts of the amount of bacteria or type of bacteria that is present in different regions of the body. For example, we have in the mouth we have firmicutes, that is a major commensal bacteria. So the commensal bacteria is highest in the mouth, followed by esophagus, then colon, then stomach, and in the vagina also we have the highest. In the skin it's the minimum. Okay. Sorry. Then comes the second one, bacteroides. So bacteroides are major in the colon, then in the esophagus, st- stomach, mouth, and minimum in the skin. Okay, we have actinobacteria. Then this actinobacteria are present mainly in the skin. Just a minute. So the actinobacteria are present minimum in the skin and followed by the stomach, and the minimal in the colon and the esophagus and the mouth. Okay, so this is how different types of bacteria are distributed among different parts of the body. Okay, so if you guys have any doubt, you can ask. Okay, so let's move forward. Now we'll move on to the receptors that actually help in recognizing different um, pathogens and different biomolecules. Okay, we have the name itself represents the pattern recognition receptors. That means they recognize different patterns. Okay, so we have different classes, like we have toll-like receptors, TLRs, rig like receptors, RLRs, or nod like receptor, that are NLRs. Okay, so toll like receptor recognize basically bacteria, fungi, viruses on surface and in endosome of cells to induce pro-inflammatory cytokines and type 1 interferons. Okay, so TLRs recognizes bacteria, fungi, and viruses that are present on surfaces and in endosomes of cells. So these TLRs can be present either on the surface of the cell or the endosomes. Okay, just 1 minute. So these TLRs basically induce the production of pro-inflammatory cytokines and the type 1 interferons for the antiviral activity. Okay, then we have rig-like receptors. These are intracellular sensors. So these are present inside the cell that can recognize RNA of the RNA viruses, and they can start producing the pro-inflammatory cytokines and the type 1 interferons to initiate their immune response. Okay, then we have node-like receptors. These are also intracellular that recognizes various pathogen-associated molecular patterns to induce the pro-inflammatory cytokines, and this is basically prediction of the IL-1 family. For the TLRs and the RLRs, these basically produce type 1 interferons, but for the node-like receptors, these majorly induce the production of IL-1 family of cytokines. Okay, so here are some of the examples of the TLRs and how they are present over the different surfaces. We have TLR4, TLR2, TLR6, TLR1 and 2, and TLR11 and 5 that are present on the surface. On the endosome, we have TLR7, 9, and 3. Okay, so you guys must remember that on the endosome we have TLR7, 9, and 3. On the surface we have TLR4, 11, 5, and 6 and 2. Okay, these have different functioning based on their production as well as on their activity. Okay, so I'm not going into that much depth, but these lead to the production of type 1 interferon or the pro-inflammatory cytokines that lead to the the activation of the NF kappa B pathway, that is the major pathway for the inflammation. Okay, then we have this dectin 1 that majorly activates the phagocytosis, the ROS production, and this inflammasome complex. Okay, so this leads to the inflammasome complex with the NOD1 and NOD2 receptors, and uh, these can actually produce the interleukin 1 beta that activates the particular immune response against the virus. Okay, so this is how these TLRs help in the production of the type 1 interferon. Okay, for the antiviral immunity, the induction of the inflammasome, and then the production of the interleukin 1 for their immune response. Okay, so this is how different TLRs function. Okay, so we'll go into a bit detail about TLRs and how they function for the production of the inflammatory cytokines and vice versa. So there are different toll-like proteins or that are present in the human, and these are basically single pass membrane proteins. Okay, so single pass means, like for example, GPCR, it's a multi-pass protein. Okay, so it has multiple 7 transmembranes that pass through the single membrane. So these are single pass, that means through the membrane only single peptide will be passing through. Okay, and different TLRs will have different domains that are present on the outside of the surface or the outside of the membrane. Okay, so there are 18 to 25 copies of a leucine rich repeat that compose each leucine rich repeat composed of 20 to 25 amino acids. So these differ in different but at TLRs. That's how there are different number of TLRs present here. Okay, then we have another component that is RP105. So this RP105 has the similar domain present towards the outside of the cell, but it doesn't have the uh, intracellular domain. Okay, so this is the TIR family. Now there are different PAMPs that are present on the pathogen that are recognized by the this TIR family or the toll-like receptor family. Okay, so for example, this TLR1, 2, 26, and TLR4, these recognize lipid and the lipid lipoproteins. Okay, so TLR1, 2 recognize triacyl lipopeptide, TLR26 recognize diacyl lipopeptide, and TLR4 recognize lipopolysaccharide that is present in the bacterial cell. Okay, then we have TLR5 and TLR11 that recognize proteins. So TLR5 recognize flagellin and TLR11 recognize unpathogenic bacteria. So this helps in recognizing the bacteria that are safe for human body. Okay, then we have TLR3. So now we have the other biomolecule that is DNA or RNA. So TLR3, 7, 8, and 9 are basically helpful in recognizing the nucleic acids. So TLR3 helps in recognizing double-stranded RNA. 7 and 8 recognize imidazoculonins and single-stranded RNA. And TLR9 basically recognize this CPG-rich DNA. Okay, so these are the different TLRs that recognizes different biomolecules. So to repeat, lipid are recognized by TLR1, 2, 2, 6, and TLR4. Okay. Then the proteins are recognized by TLR5 and 11, and DNA or RNA are recognized by TLR3, 7 and 8, and 9. Okay. So these are the different TLRs that recognizes different components. So moving on, so some of the TLRs that we have studied, for example, 1, 2, 2, 6, they become heterodimers, okay, that can recognize different ligands, and then in there can be some TLRs that are present differentially in certain cells only. Okay, so we have TLR 1 and 2 ka homodimer, sorry, heterodimer that recognize lipomans, lipoproteins, lipoic acid, and TLR 26 also. So cell wall beta glycans and zymosan from the fungi. And their cellular distribution will be monocytes, dendritic cell, mast cell, eosinophils, and the basophils. Similarly, TLR3 recognizes double-stranded RNA and poly I and C, poly IC. Okay, so these are present in macrophages, dendritic cell, and the intestinal epithelium. TLR4 recognizes LPS, lipoic acid from the gram-negative bacteria, and these are present in the macrophages, dendritic cell, and eosinophils. Okay, so likewise, we studied TLR5 recognizes flagellin, TLR7, 8, and 9 they can recognize the DNA, and these are majorly present in the dendritic cell, macrophages, and the phagocytic dendritic cells, and the eosinophil. Okay, TLR10 it is present in human only. Its function is unknown, but it is found to be present in the phagocytoid dendritic cell, eosinophil, B cells, and the basophils. 11, 12, and 13 are present in mouse only. They are not present in the human. And these can recognize, to 11 and 12 can recognize the proteins, and the 13 can recognize the single stranded RNA. Okay, especially the bacterial ribosomal RNA, and these are present in macrophages, dendritic cells, and of the liver, bladder, and kidney, as well as on the this TLR13 is present in the macrophages and the dendritic cells. Okay, so this is a bit tricky because you have to actually manage to learn the function of each of the TLR and where it is present. Okay, so out of these, TLR10 is only present in humans but not in other organisms. 11, 12, and 13 are present in mouse only, and the other TLRs are generally present in all of the higher mammals. Okay, so how they actually function, the signaling. Okay, so first, if there is any doubt, you can ask in this particular slide. Okay, so how they actually signal their thing. So TLR has two pathways for signaling, as we discussed before. We have the MyD88 dependent signaling that helps in myeloid differentiation, primary response gene is 88. Okay, so these help in macrophages, dendritic cells, and the plasmacytoid dendritic cells, that is PDCs. Okay, then we have the TRIF dependent signaling. So TIR containing adapter inducing IFN beta. Okay, so interferons producing. So these pathways basically seen in macrophages and the dendritic cells. Okay, so first for the MID D88 pathway, what happens is these TLRs directly act on a protein that can be a amplification protein or a signaling protein, that is MyD88 present in the cytoplasm. Now, this MyD88 activates IRKs, TRAP6, IKK complex, and NF kappa B. Okay, that is bind to the IKB. So these actually induces the production of IFN gamma, type 1 IFN gamma, and inflammatory cytokines like that is produced from the NF kappa B. Okay, so this is the function of the production of the inflammatory cytokines and the interferons for the antiviral activity. So this is the first molecule that takes place in the this signaling pathway, and it can further activate either IRF7, IR that is TRAP6, and the TRAP6 can further activate the NF kappa B pathway, and TRAP6 can also activate the MAP kinase pathway for the P38 gene. Okay, for its production. Then moving on to the second pathway, that is the TRIF dependent pathway. So TRIF dependent pathway also involves the production of the signal from the TLR4. For example, it is present. It activates the MAP kinase pathway and the interferon 1 producing genes. Okay, inducible genes that leads to the production of the inflammatory cytokines. Okay, so the major difference is most of the TLRs, including 1, 2, 2, 6, 4, 5, and 11, and the DNA recognizing TLRs, they function by the MyD88. But TLR4 and TLR3, okay, these two only major the functions with the TRIF activation that increases the production of the inflammatory cytokines. Okay, so this is the major difference in the both signaling of using the TLRs. Okay, so there are some other uh, inflammatory mediators that are present in the human body to suppress the immune response, to increase the immune response. These are vasoactive amines like histamine and serotonin that we studied in the last class. The eicosanoids, thromboxane, leukotrienes, and prostaglandins, peptide bradykinin, the proteins, that is interleukin 1 beta 6 and TNF alpha, type 1 interferons, type 1 interferons inducible genes, and the type 3 interferons. Okay, so these are different biochemical components that we studied in the last class. Now, how the type 1 interferons actually help in antiviral response. So the type 1 interferon helps in the adaptive immunity. So these increase antibody and CD8 T cell production and increase in the MHC1 expression. Okay, so we'll see in detail in the subsequent classes what is actually MHC1 and how it helps in antigen presentation. Okay. Then we have oligoadenylate synthase. So when ATP is actually linked to the oligomers, so it leads to the production of the type 1 interferons. Okay, that activates endoribonuclease RNase L. So RNase L can degrade the viral RNA. Okay, we have another P1 kinases. So it activates the EF12, so it inhibits the translation of the proteins of the virus. Then we have MX proteins that increases the resistance to virus, that is particularly in influenza. And then this type 1 interferon activates NK cells that kill the virus infected cells. Okay, so this is how type 1 interferons function in different context of the particular body. Okay, so any doubts in this particular slide? Okay, so let's move on. So, uh, in some of the cases, what happens is there is polymorphism. Okay, so polymorphism basically is a genetic term that is seen in population. Okay, so this is basically in terms of the population genetics. Okay, so as for example, it is a gene here in a particular population, G is present. Okay, but due to mutation or in any of the things that takes place due to the environmental conditions and any of the reasons, this G in half of the in the new population is replaced by A. Okay, so this particular thing leads to a particular gene in a population, and because there is a single base pair change in half of the population, that means the population can be prone or superior in case of the phenotype of that particular gene. Okay, for example, considering the fact that this gene can be either insulin producing gene. Okay, so I'm giving you an example. If this G to A transition is basically helpful in maintaining a resistant for the population to become diabetic. So what will happen? This G, the population that have G, would be actually prone to diabetes. Okay, then we have A that is resistant to diabetes. But in both the cases, the individual are normal. They do not have diabetes at all. But because of this presence of this one nucleotide change, there is difference in the insulin structure or in the splicing and the production or the expression of insulin. So that makes the G as prone to diabetes and this population that have A that is resistant to diabetes. Okay, so this is called as polymorphism and that distinguishes between population to population. Okay, so now what happens is this TLRs also show some of the क्या कहते हैं some of the polymorphism so that they make those particular organisms susceptible or resistant to the disease. Okay, for example, in TLR2, we have seen that T597C or R5753Q. So, this is a protein and this is a nucleotide. So, in this case, this is associated with a protective effect against leprosy and predisposition to tuberculosis. So they can, these individuals are more protected against leprosy. So they do not suffer leprosy, but they are more prone to tuberculosis. Okay, and they are more prone to staphylococcal infections. Okay, so because they have some structural change that cannot recognize the staphylococcal pathogen, so they can be easily infected by the staphylococcus. Okay, then we have TLR4. So it includes D299G and T399L. That means it has increased systemic inflammatory syndrome and septic shock, decreased risk of atherosclerosis. Okay, so these patients have increased risk of systemic inflammatory syndrome and septic shock, but decreased risk of atherosclerosis. Okay, then we have TLR5. That means a dominant negative stop codon mutation. This increase the susceptibility to pneumonia caused by Legionella or pneumoniella, and negatively correlated with the Crohn's disease. Okay, so Crohn's disease, they are protected, but increase susceptibility to pneumonia by the pathogenic organism. Similarly, there are some more receptors like MAL that have the polymorphism is S180L. This help in against tuberculosis, severe sepsis, malaria, and there no effect on rheumatoid arthritis. Then we have IRF5. So these are the nucleotide changes that SMP that leads to the SLE. Okay, IRAK4 IRAK4, several SNPs including C877T. So they actually are prone to Streptococcus pneumonia and Streptococcus aureus infections, decreased inflammatory response to LPS, and increased IgE concentration in serum of patients with CR5 and CRS and asthma. Okay, then we have MID88, that is a major signaling molecule for the TLRs. So the minor variant in 3' UTR, this increases the decreased antibody response to measles. So this is how different polymorphisms basically help in maintaining a particular individual prone to a particular disease or protects against the infection from the other diseases. Okay. So if you guys have any doubt, you can ask. Okay. So let's move to the questions. What is the primary function of lysosome in combating pathogens? So what can be the correct answer? It would be lysozyme. Um, Okay, so lysozyme basically helps in degrading the cell wall of the bacteria. Okay, so yes, the correct option is on answer number two. So it hydrolyzes bacterial cell walls, leading to bacterial lysis. Okay, so let's move on to the next question. What is the primary function of TLRs in the immune system? So what can be the correct answer? Okay, so yes, the correct answer is option 3. That means it will be able to rapidly detect the diverse array of pathogen associated molecular patterns that triggers the innate immune responses. Okay, so this is the major function of the TLRs. So these are involved majorly in the innate immune system. Okay, so let's move on to the next question. Which known polymorphisms of IRAK4 affect the TLR signaling? So, what can be the correct answer? Okay. The correct answer is option 1. So, polymorphism introduces top codons at 287 and 293 position that increases the susceptibility to gram positive bacteria. Okay, so this was the polymorphism. So, let's see. Okay, so it is not mentioned here, but yes, the correct answer is option number 1. Okay, so let's move on to the next question. What is the consequence of antibiotic treatment on commensal bacteria in the microbiome? So, what can be the correct answer? Okay, so the correct answer is option 2. So, it leads to dysbiosis. Okay, so dysbiosis is basically the imbalance in the microbial community. Okay, so this is the imbalance in the microbial community caused by the antibiotic treatment. So it will actually kill both type of bacteria, either it is good or bad. Okay, so this is the major term, the major meaning of the dysbiosis. Okay, so let's move on to the next question. What are some characteristics of innate immunity that make it essential for survival? So what can be the correct answers? 5 and 7. Okay. So let's see the options. Acts as the first line of defense against microbes. Yes, this is correct. So this is the major property of the innate immunity. Then we have requires prior exposure to pathogens for effectiveness. No. Okay, this is the major property of the adaptive immunity. Innate initiates an immune response within hours of exposure. Yes, this is correct. Provides long term immunity. No, because this is a short term. Okay, so for the initial infection by the microorganism, this is takes place, and it comprises an ancient and evolved system. Yes, this is also correct, because the sponges that are the early organisms also show the innate immunity. Okay, then adapts quickly to new pathogens. So no, because new pathogens are also recognized in the same way like the old pathogens. Okay, it recruits effector cells to fight infections. Yes, this is also correct. Then influences health through the microbial gene pool. Yes, this can be the correct option. Okay, so the correct answers are 1, 3, 5, 7, and 8. Okay, why? Because this innate immunity actually helps in recruiting the microbes so that they can actually help in the antimicrobial pathway. Okay, so any doubts in this question? Okay, so let's move on to the next question. MBL belongs to which family of immune proteins and what is its primary function? Okay, so MBL basically stands for the mannose binding lectin. Okay, so this mannose binding lectin belongs to the lectin family, and it binds to the surface sugar molecules on the pathogenic surface. Okay, so the correct answer is option number 3. Okay, so any doubts in this particular question? Okay, so let's move on to the next question. Select all statements correct that correctly describe the biochemical and chemical barriers involved in the innate immunity. So, what can be the correct answer? let's see the options. Lysozyme breaks down peptidoglycan in the bacterial cell wall. Yes, this is correct. Complement proteins act only as a part of the adaptive immune response. No, complement also acts as a part of the innate immune response. Mannose binding lectin bind to the sugar on pathogen surfaces. Yes, this is correct. Alpha defensins are antimicrobial peptides produced by the gut and lungs. Okay, so this can be correct. NK cells directly from part of the chemical barrier stored in the laminar bodies? No. Okay. CRP is a pattern recognition receptor on immune cell surfaces. Yes. Sorry. No, this is also false. Then cathelicidin is an antimicrobial peptide stored in the laminar bodies. Yes, this is correct. So cathelicidin is like defensin. It is also produced in response to the pathogens. Okay. Then histactins are antimicrobial chemicals present in the oral cavity. No, this is false. So the correct answer is okay. Sorry, this is correct. So one, three, four, 6 and 8. Okay. So 6 is correct. Then 7 is not present in the laminar bodies. Okay, so seventh one is wrong. But CRP is a pattern recognition receptor that is basically present on the that is present in the liver. Okay, so it acts as a pattern recognition receptor that helps in recognizing the pathogen in the liver. Okay, so any doubts in this question? Okay, so let's move on to the next one. The term PAMPs referred to. So what can be the correct answer? Okay, so the correct answer is option 2. So PAMPs refer to pathogen associated molecular patterns. Okay, and these are present on the pathogens that actually help in differentiating those pathogens from the normal microbes. Okay, so that is why these are the molecules that are shared by the different pathogens. So the correct answer is option 2. So let's move on to the next question. Which follow family of pathogen recognition receptors is primarily responsible for sensing the RNA viruses? So what can be the correct answer? Okay. So yes, the correct answer is option 3. That is RIG-like receptors. So RIG basically stands for the retinoic acid inducible gene. Okay. So inducible gene 1. So these actually sense the single-stranded RNA of the viruses. Okay, and that's it. So these are the rig-like receptors or the RLRs. Okay, so let's move on to the next question. What is the significance of the TIR domain in toll-like receptors? So what can be the correct answer? Okay, so the correct answer is option one. So it helps in basically the downstream signaling from the particular receptors. Okay, so what happens is this is the outer membrane domain, that is the leucine rich domain, and this is the TIR domain. So it actually helps in increasing the downstream signaling after the PAMPs has been bound. Okay, so let's move on to the next question. Which of the following statements are correct? So what can be the correct answer? Okay, let's see. Innate immunity evolved 450 million years ago since the origin of the sponge. So sponges actually evolved 700 million years ago. Okay, so this statement is false. Adaptive immune system evolved 700 million years ago since the first jawed vertebrates. No, this is 415 million years ago. Okay, so this option is also false. Innate immune genes evolve slowly while pathogens evolve rapidly. Yes, this is correct because innate immune genes help in recognizing the pathogens. And whenever the pathogens evolve, these genes actually become short of recognizing those pathogens. Okay, so dermis gives strength, skin strength, and pliability to resist abrasions that could introduce the pathogens. Okay, so yes, this is correct. Then sebum lowers the pH of the skin to a level inhibitory to many bacteria. Yes, this is also correct. So the correct options are C, D, and E. So the correct answer is option number 4. Okay, so any doubts in this question? Okay, so let's move on to the next question. Which among the following is not an antimicrobial peptide or protein? So what can be the correct answer? Okay, so lactoferrin, lysozyme, serum amyloid A, antitrypsin, ceruloplasmin, and azoceride, azuro Cytidine. These are all antimicrobial peptide or proteins, but insulin is not an antimicrobial peptide or protein. Okay, so let's move on to the next question. Which among the following is not a reactive oxygen or nitrogen species? So, what can be the correct answer? Okay, let's see. So the correct answer is option number six. Okay, nitric oxide is an nitric oxide is the nitrogen reactant nitrogen species. This is also a ROS. This is also a ROS. This is also an ROS. Similar to this. Sorry, this is an RNS. Then nitrous oxide is not a RNS. Okay, we have H2O2 and nitrogen dioxide. These all are ROS or RNS, but nitrous oxide are not an oxygen reactive species. Okay, so let's move on to the next question. Which among the following TLR is only present in mice but not in humans? So what can be the correct answer? Okay, so the correct answer is option 11. So TLR11 is only present in mice but not in humans. Okay, so let's move on to the next question. Which TLR utilizes MyD88 independent signaling pathway? So what can be the correct answer? Hmm. So the correct answer is option 1, that is TLR3. So TLR3 functions with MyD88 signaling pathway. Okay, the other two works with the signaling pathway. Okay, so let's move on to the next question. Which of the following pairs related to the receptor and their ligands are incorrect? So what can be the correct answer? Yes, the correct answer is TLR8. It recognizes single stranded RNA. Okay, so TLR8 7 both recognizes the DNA or the RNA. That means nucleic acid. Okay, so they do not recognize proteins. So option 2 is incorrect. Okay, so let's move on to the next question. Which of the following TLRs can sense single stranded RNA viruses? So what can be the correct answer? Yes, just now we discussed. both 7 and 8, they can recognize single stranded RNA. Okay, so they both have the capacity to recognize the single stranded RNA viruses. Okay, so let's move on to the next question. Which among the following TLR is not located in endosomes? So what can be the correct answer? Okay, so the correct answer is option 1, that is TLR4. This TLR4 is not located on the endosome, this is located on the surface of the. Okay, so rest of the two are located both on the surface as well as the endosomes. Okay, so let's move on to the next question. So this was the last question for today. If you guys have any doubt regarding any of the components of the PPT, you can ask. Mhm. Okay, if no doubts, then we'll close the session for today. Thank you guys for joining in. We'll see you next week. Thank you.